everybody. I'm going to spend a little bit of time and tell you a bit about how we develop and grow these nutraceutical crops that we use as sources of the bioactive molecules that are involved in our products from Kemin. One of the first things that we do when we look at a new product is we need to ask ourselves what consumers really want, what their expectations are. And there are two clear areas. The first one is clean label. It's typically non-GMO sourced, ideally sustainably grown, which includes real environmental and social responsibility in the production of that particular crop and the molecules involved. And in some cases, applications for organic production as well. In the health benefit area, there are three key components there. One is the potency of the product. Obviously, it has to be able to be potent enough to have a physiological response. The consistency of the product has to be there, so every single purchase essentially is the same. And of course, there has to be efficacy per dose as well. At Kemin, we see plants as molecular factories. They really are tremendously sustainable sources of a whole range of molecules. And the beauty of them is that they use sunlight as energy. They sequester carbon dioxide from the atmosphere. And they use water in their growth cycle to produce, in many cases, some very complex molecules. In the example I'm showing here, it's rosmarinic acid. And in many cases, plants will produce these target molecules at up to 10% of their dry weight. And when you think that 80% of the dry weight is plant cell wall, for a plant to dedicate 10% of its dry weight to one more molecule is tremendous. It doesn't leave much room for anything else. Oxygen is also a byproduct, which is good, and obviously we're able then to return the captured carbon from the atmosphere back to the earth, again, which is a very useful way to sequester and recycle carbon. In terms of how we use plants, there are three basic areas that we can derive nutraceutical molecules from plants. I'm going to talk a little bit about each one individually, but they include wild harvest, just taking what Mother Nature has to offer and collecting that from the wild. On the other extreme is through genetic engineering and improvements via genetic modification or genetic engineering. The avenue that we prefer to play with is the improvement via plant breeding, and that's conventional plant breeding through crossbreeding. So I'm going to take just a few minutes to try and explain what those terms are and how they differ from each other. In the case of wild harvest, this is biomass that's collected from wild plants growing out in a wild environment. The two photographs in this slide are actually examples of rosemary growing wild in southern Europe. Oftentimes, companies will send out collection crews onto slopes, hillsides, or wild areas to collect plant tissue. And there are several pros and cons with doing this. We'll just address some of these and then compare them to the other two sources. The first pro, obviously, is that it's natural. It's coming right out of the wild. Typically, it's a cheap source of biomass. Harvest crews and extraction companies will typically get a license from a local government to be able to go onto the land to do this. Oftentimes, they're not allowed back onto the land for up to 2 years, and that's primarily because of the negative impact that this kind of clear-cutting or wild harvest has on the environment. And of course, the other pro there is it's clean label. It's coming right out of a natural environment. The con side here, obviously, there are three components there. The first one is that each of these plants will have been derived from seed. So an individual seed will have fallen on the ground, it germinates and grows, and that makes each individual plant genetically different from each other, which then results in quite a wide variation in the accumulation of target molecules. That often leads to lower potency of the entire biomass that's collected, and then an inconsistent level of the bioactive molecules in a particular extract. And because companies are not allowed back in the area for anywhere up to 2 years, at Kemin, we don't believe that that's environmentally sustainable. It doesn't make sense to use a natural product if there is impact, a negative impact to the environment when you actually are able to produce that particular product. On the other end of the spectrum, there's the use of genetically modified crops or crops that have been developed through genetic engineering. And these plants, typically referred to as GMOs, contain foreign DNA from other species, and that DNA is introduced. It allows pathway engineering for the hyperaccumulation of these molecules. And perhaps the most well-known example of this is golden rice, that accumulates very high levels of beta-carotene ideally for the production of vitamin A. And this was introduced several years ago for malnutritioned children to try and provide a readily available version of vitamin A. The pros here, obviously, is that because of genetic engineering and the hyperaccumulation, it's highly potent. It can be grown under sustainable conditions or good agricultural practices that provide a sustainable approach to crop production. Because of the hyperaccumulation, it can be low cost. And because of the genetic engineering, there's an opportunity there to produce complex bioactive molecules. On the con side, it is not a clean label. It is GMO. And today's consumer general distrust of this particular technology means that it's not always necessarily available to everyone. At Kemin, we like to use an entirely different process of purpose-bred crops. And these are plants or crops that have been optimized for the accumulation of specific molecules through conventional plant breeding or cross-pollination. And there are some examples there of making individual crosses between plants that hyperaccumulate so that we can generate populations from which then to select out plants that really do a fabulous job at accumulating these molecules. The pros here are that they're natural because it's just conventional plant breeding. The same type of technology used on most food crops that use conventional plant breeding. It's potent because through the process of plant selection, we can identify individuals that, again, hyperaccumulate. It's clean label because it's conventional plant breeding. We can grow under sustainable or good agricultural practices. And at Kemin, we use a process that allows us to continually improve these genetics so that every 3 or 4 years or so, we introduce new improved lines that continues the improvement of the efficiency of the production of the molecules and obviously the subsequent extraction. One of the cons of this is development time. It can often take anywhere from 7 to 10 years to develop a new hybrid. And so the process is slow to begin with, but as you develop a pipeline of new hybrids or new clonal lines, the development can be fairly rapid after that. So at Kemin, in order to do that, we've assembled a team of experts, really a global team of experts, and they include 5 specific sections. The first one is breeding and selection, and that particular group's job is to create genetic variation through conventional plant breeding practices. Our second group, the biochemical screening and phytochemistry group, is based at our facility in southern China, in Zhuhai. And their job is to measure the genetic variation and provide that data back to the breeding and selection group so that we can select individual plants that have chemotypic profiles that we're interested in then to scale up. Working closely with breeding and selection is the research agronomy group. Very often we have to develop entirely new cropping systems for these plants. Many of the species that we grow would typically be seen in a terracotta pot on a windowsill, and we have to translate that now to hundreds, if not thousands of acres under just outside general field conditions. And that requires an entirely new type of cropping system development, everywhere from planting the crop, the control of pests and weeds, and all the way through to harvest and post-harvest of the selected tissue that we want to extract. Beyond that, then, we have a phytochemical extraction group. In fact, we have 2, one in the U.S. and one in China, and their job's to develop extraction processes that optimize and concentrate these bioactive molecules. And then working in direct partnership with our growers is our field agronomy services group. That means we have agronomists working with our growers, our contract growers all over the world, every day on farm to make sure that they adhere to some very strict production practices. And that team works very closely together. It's a continuous process, a fully vertically integrated process for the production of these highly bioactive molecules. We've also invested in quite a few facilities. In Iowa, we have a molecular breeding lab that allows us to do DNA extraction. It allows us to follow the movement of specific genes through a breeding program, allowing us to select for flower-type traits, for example, in seedlings well before they would even flower. And that's a fabulous technology. Again, it's conventional plant breeding, but it allows us to monitor the flow of these genes through a breeding program. We have a micropropagation lab that allows us to take an individual plant and generate up to 80,000 identical plants within a very short period of time, which then can be used for conventional propagation in a greenhouse following that initial move. We have a 10,000 square foot greenhouse complex in Iowa where we do most of our primary plant breeding, and then we have a 500-acre research farm that the research agronomy group used to develop new cropping systems. In Texas, we have a similar 10,000 square foot greenhouse. It's primarily dedicated to our marigold breeding program. And then in Brazil, we have a small 2-acre research farm that allows us to continue breeding year-round. So selections made in the summertime in the northern hemisphere can be taken to the southern hemisphere for further selection and then back to the northern hemisphere to continue that cycle. And that allows us to move as efficiently as we possibly can.